Okay, section 2, we're going to talk about the structure of a virus and how they copy themselves or replicate, which is the fancy biology term. So I need you to know all the vocab. I need you to know the sizes and shapes that viruses come in, not every single one, just generals, and then how do viruses reproduce. This is going to be the hardest part. But once you understand it, you'll be okay. Don't worry about it. So viruses differ on a couple of things, mostly their shape and their strategy on how to get into a cell. Right? They are pretty simple organisms. They're not organisms. They are simple. They're genetic material surrounded by a protein coat. That protein coat is called a capsid, kind of like a capsule, right? Like when you take pills, that's a capsule. Well, they contain the genetic material. Now, the capsids can be lots of different things. Like over here, we have some that are just like a big old cylinder. We have some that has this really cool, I think it's icosanoidal shape, and even creepier ones like this one, which looks like a weird spider-looking thing called a bacteriophage, right? All different shapes and sizes, and always remember that form and function go together. Why do things look like this? Well, because it's going to tell us a little bit about their function. Well, this one has all these little spiky things around because it's going to stick to something. This one is the shape because it likes to burrow down. Notice how this one has a kind of similar shape too, but has these neat little tail fibers things. Well, these little fibers are like legs. They walk around on a bacteria. Now, viruses, they don't have like cell membranes. They aren't cells. They're smaller than cells, if you remember from last section. So how do they operate? What do they do? Well, because they lack organelles, they have to pretty much steal everything they got from other things. How do they do that? Well, they invade them, all right? And they just use whatever the cell they're invading had. They're like secret agents of biology world. They just go in, take over the whole place, and what are they trying to do? Well, they're trying to make copies of themselves, right? Now, one of the viruses I need you to focus on in particular is a thing called a bacteriophage, right? And that's kind of confusing because it has the bacteria in it, and it makes you think it's a bacteria, but it's not. It's a kind of virus that is specific for bacteria, right? If you remember, that word phage means to eat. So the word bacteriophage literally means something that eats bacteria. And that's what these viruses do. How they do it is they have their genetic material here, in this case DNA, and all this in black is the proteins. This part can walk around on the cells, and it will eventually inject the DNA into the cell, which will then go and take over that cell. Now, while the bacteriophage is not going to harm a eukaryotic cell because it's a bacterial virus, it can still exist inside of a thing like a mammal, right? Because even inside mammals, there are bacteria in there, and viruses, they want to infect and attack and replicate themselves, and they do that by attacking bacterial cells. Now, eukaryotic cells are a little bit different, right? Viruses are changed based on how they enter the cell. Some are going to force their way to the cell, and some are going to be intentionally uptaken by the cell through endocytosis. Really doesn't matter how they get into the cell, just that they get into the cell, because the end result is the same. That is that the host DNA is going to be hijacked by the viral DNA, and it's going to use all of the proteins, all of the organelles to do the virus's bidding, which is really creepy when you think about it, because it's not a cell, it's not alive, but it has this much power over a living entity, right? And the two types of infections that can happen through a virus is the lytic and the lysogenic cycle. Now, lytic, I want you to tie in your head, lytic, lyse, and lyse means to break apart, right? Like hydrolysis, water breaking, right? In the lytic infection, what's going to happen is the virus is going to get its way into the cell, merge its DNA with host DNA, and have it create more and more viruses inside that cell until it literally explodes the cell, which is really cool, by the way. Yeah, it's pretty neat. So nothing else I know of can do that, but in the lytic infection of a virus, they can and do that. In the lysogenic cycle, it's kind of the same, right? But I want you to think about this word, the genic, that means to produce something. Well, in the lysogenic cycle, the virus is more playing like a long game, if that makes sense. What it's doing is trying to embed itself into the DNA of the host and then let the host copy itself into another organism. That's kind of like it's getting two infections for one, if that makes sense. The more times the infected cell reproduces, the more free infections the virus gets. they combine themselves with the host DNA, and at any time they can return back to going through a lytic infection and cause that cell to rupture. So really, really insidious, really gross. These are not unique to each other. In fact, lysogenic will lead to a lytic cycle eventually, because remember, this is how they go and infect other cells. But just because there is a lysogenic infection doesn't necessarily mean it takes place. Sometimes the virus immediately goes to the lytic infection. Sometimes it just does the lysogenic infection. So that's what we're looking at here. Over here in the lytic cycle, the virus is going to put its DNA into the cell, and that looks to be a bacteriophage, so I'm going to assume this is some kind of bacteria. And this is the protein coat. Remember, it's not important. That just goes away, right? What's important is inside the cell, this DNA is forcing the bacterial cell to now make more and more viruses and assemble them. And it's going to assemble them until such a point where it breaks the cell apart and then release them. And what are all these other viruses going to do? Well, they're going to restart the cycle up here. Now, the lysogenic cycle is the lytic cycle, just like a longer game, like a chess game. So the bacteriophage still invades the bacteria, but instead of immediately starting production of more viruses, it incorporates its own DNA. That's that little red strand right here. So red strand, into the larger bacterial DNA complex. And remember, bacteria can divide pretty quick, sometimes as fast as 15 to 20 minutes. So within an hour or two, we can have lots of infected bacterial cells. Whenever the virus is ready, it can just resume the lytic cycle and destroy the cells and create vastly more viruses than from just one lytic cycle alone. So it's kind of scary the way they work and how efficient it is, and really eerie and creepy, but that's what makes them really neat. Even though they're not living, they don't have brains, they don't have cells, that they can do all this and do it very well, as we can see with lots of different viral infections. Okay, so in review, what is the purpose of the viral capsid? And what kind of viruses are the eaters of bacteria? What is their actual name? And be able to compare and contrast the lysogenic and lytic cycle, right? They're very similar. One leads to the other, but know those like the back of your hand. And always remember, lytic means to break something, lysogenic means to like make something, so that can be a hint towards which one is doing what. If you have any questions, please email me. Thanks for watching. See you next time when we go over the third section. See y'all later.